Assalamu alaikum warahmatullah. Today's topic is central dogma. Today we will discuss about the central dogma. It is a topic taken from the genetics of class 12th. Now, what is central dogma? The unidirectional flow of information from one nucleic acid to another nucleic acid for the synthesis of proteins is called as central dogma. The term central dogma was coined by Francis Crick. The central dogma has two steps. One is called as the transcription and second one is called as the translation. Transcription simply means the synthesis of the mRNA from the sense strand of DNA, and the translation simply means the synthesis of the proteins from the messenger RNA. Before we proceed to the central dogma, we will discuss about the topic genetic code. It's necessary to know about the genetic code before we discuss about the processes of transcription and translation. Now, what is a genetic code? As all of you know that the whole of the information in a cell is packed in the DNA. The DNA is the genetic material. It stores all the genetic information of a cell, and the DNA helps in the synthesis of the proteins. It is the protein that expresses the characters within the cell. It is the protein that controls the different metabolic activities within the cell, and these proteins are synthesized inside the cytoplasm on the surface of ribosomes, and the information for the synthesis of proteins is found inside the nucleus in the form of DNA. Thus, DNA passes the information for the synthesis of proteins first to the messenger RNA in the form of code words, and that code word is called as the genetic code. And later on, the proteins are synthesized from the information of the messenger RNA inside the cytoplasm on the surface of the ribosomes. The term genetic code was coined by a physicist named as Gamow. The genetic code is defined as the relationship between the sequence of nitrogenous bases in DNA or messenger RNA and the sequence of amino acids in a protein. The first genetic code discovered was UUU. It codes for the amino acid phenylalanine. It has been various times asked in the competitive examinations, NEET examinations. The first genetic code discovered was UUU, and it codes for the amino acid phenylalanine. Thus, the information for the synthesis of the DNA is packed inside the nucleus, and the synthesis of the proteins takes place inside the cytoplasm. The DNA passes its information to the messenger RNA, and later on, this messenger RNA passes from the nucleus inside the cytoplasm for the synthesis of the protein. DNA is composed of 4 nitrogenous bases, and it is said that there are more than 20 amino acids in a cell. However, only 20 amino acids gets coded to form the different types of the proteins. It is yet a challenge to the scientists how many proteins are synthesized inside the cell. Just like we have 26 alphabets A to Z, but there are infinite words that are developed from these 26 alphabets. In the similar way, there are large infinite number of the proteins that are synthesized from these 20 different amino acids. The information in the DNA is packed in nitrogenous bases, and there are 4 nitrogenous bases in the DNA, and if one nitrogenous base code one amino acid, then only 4 amino acids are coded. If these nitrogenous bases are arranged in the form of doublets, thus 16 amino acids are coded. But once these nitrogenous bases are arranged in the form of the triplets, 64 codons are developed, and these 64 codons code for the 20 different amino acids in a cell to form the different types of the proteins. So we can form the genetic code in this way. This is UCAG, 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 UCAG. The genetic code is a triplet code. That means it's composed of 3 bases. We will develop these genetic codes in this manner: U, U and U, 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 C, U, U, A, U, U, G, U, C, U, U, C, C, U, C, A, U, C, G, U, A, U, U, A, C, U, A, A, UAG, UGU, UGC, UGA, UGG. So in this way, we will develop 64 codons that codes for the 20 different amino acids in a cell to form the different types of the proteins, and these different types of the proteins controls or regulates the different metabolic activities of the cell. Okay, now that means the DNA passes its information to the messenger RNA in the form of the genetic code, rather we call it as code words, and there are 64 different codons that codes for the 20 different amino acids to form the different types of the proteins inside the cell, and these different types of the proteins regulates or controls the different metabolic activities of the cell. Okay, now properties of the genetic code. The genetic code has certain properties. First property, the genetic code is a triplet code. That means it's composed of 3 bases: U, 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 it's a codon; U, U, C, it's a codon; U, C, U, it's a codon. So that means genetic code is composed of 3 bases. Second property, the genetic code is comma-less. There is no comma in between the nitrogenous bases. Third property, the genetic code is universal. Universal means the same genetic code codes the same amino acids in all the organisms. Fourth, the genetic code has an anticodon. For example, UUU, it is a codon, it has anticodon AAA. UCU, it is a codon, it has anticodon AGA. Recently, the anticodon is also called as NODOC. NODOC. CODON is codon. CODON. Anticodon also known as NODOC, NODOC. The genetic code has polarity. It has 5-3- polarity. That means we must read the genetic code in 5-3- polarity. It's like this. It is UCC. We cannot read it as CCU. It has a polarity, 5-3- polarity. Next property, the genetic code has an initiation codon. Initiation codon is that codon that starts the process of protein synthesis. The codon that starts the process of protein synthesis is known as initiation codon. Initiation codon is usually AUG, UG. It codes for the amino acid methionine. However, in bacterial protein synthesis, the GUG also acts as an initiation codon that codes the N-formal methionine. Next property, the genetic code has a termination codon. Termination codon means that codon that stops the process of protein synthesis. It was earlier found that there are 3 codons out of 64 codons, namely UAA, UAG and UGA, that does not code for any amino acid. They were previously called as nonsense codons, but later on it was found that these 3 codons help in the termination of protein synthesis. So they are known as termination codons or stop signals. Thus the genetic code has the termination codons, and these termination codons include UAA, UAG, UGA. Next property of the genetic code is the genetic code is degenerative. The genetic code is degenerative means one amino acid have more than one codon. There are only 20 amino acids, but there are 61 functional codons. That means one amino acid have more than one codon. One amino acid gets coded by more than one codon. That is, genetic code is degenerative. Next property, the genetic code is non-overlapping. Non-overlapping means, for example, we have UUUCA. It's not like this. UUU is one codon and UCA is another codon. This one is overlapping. Non-overlapping means UUU, UCA. This is separate and this is separate. That means genetic code is non-overlapping. However, it has been found in certain bacteriophages that there are overlapping codons. Next property, the genetic code is non-ambiguous. Non-ambiguous means one codon codes only one amino acid. For example,